Okay, it's 2 o'clock, so I think let's start the webinar. So let's go into GCLP and start with what GCLP is and why it exists. Put simply, GCLP is a set of standards for the labs that perform analysis or evaluation of clinical trial samples. Now, the standards have been designed because this work needs to be done in compliance with all the rules that apply specifically to clinical trials activities. The GCLP rules were produced because GCP hardly touches on lab work. Also, GLP, good laboratory practice, applies to non-clinical health and environmental studies, so including work conducted in the lab, in greenhouses and in the fields. So GLP doesn't apply to clinical samples. So although GCP and GLP have elements, they don't really cover what GCLP does. So the history of GCLP, 2002, Barker, now RQA, which is the Research Quality Association, published its GCLP guidance. A few years later, 4 years later, WHO published its own version, which was very heavily based on the Barker guidance. Then a few years later, you can see there were some other set of guidances, all broadly the same. One 2008 in India, 2009 MHRA's guidance for the UK, and in 2009 guidance in the US that's mandatory. Oh, someone's got their microphone on. Guidance in the US that's mandatory for labs working on DAIDS projects. In 2012, the EMA produced its reflection paper for laboratories that perform the analysis or evaluation of clinical trial samples, and that is really what this webinar is going to be based on. It's the main guidance for labs performing activities for clinical trials in Europe, but it's also used as a reference for a lot of labs outside Europe as well, which is why we're going to base the webinar on that. It's important, though, to remember that GCLP, it's not a law in the way that GCP is. It's just the legal basis for analysis of clinical of samples from trials so that they support GCP. Essentially, GCLP are the procedures you follow in a lab. GCLP are the procedures you follow in a lab so that the rules of GCP are met. So let's start off by talking about the facilities you're going to need. The labs will need adequate space for the volume of work, and the design of the facility needs to provide an appropriate degree of separation of the different activities to ensure the work's going to be conducted properly. There needs to be enough suitable accommodation for samples, reagents, paperwork, and I'm going to keep going back and back about documentation because documentation is very, very important in GCLP. So you need to store the paperwork, and you also need to be have consideration to providing specific areas for the receipt, tracking and storage of samples. So for those of you who attended the GDP webinar a couple of weeks back, you'll see there are quite a few parallels in GCLP, and for those who attended the GCP one, you'll also see there's some parallels there as well. The environmental conditions need to be appropriate for the work that's being performed to ensure that the data is going to be reliable. And it's important to ensure that the arrangements for sample handling are such that the sample integrity is maintained and cross-contamination isn't going to be an issue. Now, I've also already talked about the facilities being suitable that different operations can be separated, but that separation can either be done physically or through time, because it may be that the facilities aren't large enough for them to be physically separated, so phasing different activities at different times can provide the separation you need. There need to be procedures covering waste storage, collection and disposal, and that needs to be in line with local regulations. And where relevant, it's also a very good idea to have decontamination procedures already in place for facilities and equipment in case they're needed. Also think about security. You need to be able to ensure that samples, equipment and data can't be tampered with by anyone unauthorized. So let's look at the equipment. It sounds obvious, but it needs to be fit for purpose. It should be regularly maintained by someone who's suitably qualified to do it, and the maintenance needs to be documented. Now, before equipment can be used on clinical studies, and again, I'll keep saying this, I'll keep saying things that need to be in place before you actually start any clinical trial work. So before the equipment can be used on clinical studies, it needs to be certified as fit for use. Now, this requires an appropriate level of user acceptance testing to show that the equipment is fit for its intended purpose. What this level is going to depend on the equipment and what it's being used for, but the lab will need to justify the testing regime it chooses. So the testing should be documented again, and the records need to be retained. There should be a clear date on which the equipment was judged fit for use. It should also be possible to demonstrate that the equipment remains fit for use on an ongoing basis. So to achieve this, standard procedures need to be in place for cleaning, maintenance, calibration. You can use manufacturers' manuals as the basis for this process, or it can be done through standard operating procedures. But either way, you need to keep records of the way that these activities have been performed. A risk assessment can help with determining how often you need to do calibration. So ask yourself questions like, what's going to happen if an analyst discovers that equipment isn't working on a particular day? Will you be able to identify how many samples have been processed since the last time the equipment was checked? So how many samples could have been potentially affected? What impact is that going to have on the study? And how much will it cost to rerun all of the affected tests, not only in terms of money, but also in terms of potential damage to the lab's reputation? Use of equipment inventories is really helpful. And these should include things like serial numbers, location of the equipment, the manufacturer, service frequency, provider, related standard operating procedures, and also who is responsible for the maintenance of that equipment. Okay, this is an important point. When equipment's out of service, you need to have some sort of process in place to make sure it can't inadvertently be brought back into service. So it can't inadvertently be used by staff. And you need to ensure that any impact to a clinical trial of this thing being brought back into service is dealt with in a very timely manner. Finally, the EMA specifically requires validation of computerized systems. Automated analytical systems are pretty common in GCLP labs, but they do need to be properly and regularly validated. So now, roles and responsibilities. The individuals involved in analysing or evaluating samples from clinical trials must be appropriately educated, experienced and trained, and procedures need to be put in place to ensure that this is so. They also need to be fully aware of their roles and responsibilities on a trial. They'll need GCP training that's appropriate to their role. GCP refresher training is usually done periodically on a 2-year cycle to ensure that people stay up to date with any changes in regulations and current expectations. They'll also need training in the technical requirements of their jobs. So that includes specific training to ensure they're competent to carry out explicit techniques required by the clinical protocol, etc. All the training should be documented in a training record, which should include any relevant experience gained in previous employment or education, as well as that gained in the current post. A record can be in the form of a CV, but if you're going to use a CV for this purpose, it won't be the same kind of CV as people use for job applications, because job application CVs are more sort of about self-promotion. These are purely a document recording things like society memberships, previous experience, qualifications, academic papers. So it's quite a dry, factual document. In the training record, you should also include any certificates or evidence of courses, conference attended, things like that, as well as a record of any in-house or on-the-job training. A signed copy of the job description could also be retained there. And this is a good indication that the individual knows what their responsibilities are. Ideally, training records should be reviewed on a periodic basis by lab management just to make sure that they're still current and relevant. And this review should, yep, you guessed it, be documented. This could form part of an organization's performance review process. When people leave the organization, it's management's responsibility to ensure that a copy of this training documentation is retained for the records. So now let's look at roles and responsibilities split into 5 areas: lab management, analysis, QA, reporting, and archiving. So let's start with lab management. You need to ask yourself, are there enough resources to carry out the trial work in a timely manner? The resources need to be identified and allocated before any work starts. So are there sufficient members of staff? Are they all properly trained? Do all the staff involved in the clinical analysis have up-to-date job descriptions? And there should be a named individual overseeing the analytical work. And they're probably going to be called something like an analytical project manager. The naming isn't important, the role is important. Now, their role is to ensure that everything is done in accordance with the protocol, standard operating procedures, contracts, etc., etc. So QA, independent quality assurance activities ought to be in place to ensure data integrity and to safeguard any subject rights under GCP. So there must be a quality assurance program in place with designated personnel. Management has a responsibility to ensure that this is the case and to ensure that the activities happen in accordance with the applicable regulatory requirements. How labs do it depends very much on the scale and the scope of the work they're doing, as well as the resources they've got available. But whatever the QA activities are, they need to be documented in standard operating procedures or policies. And it's a good practice to maintain audit schedules to indicate planned and performed activities. The person who does the audit, or people doing the audit, need to be independent of the work that's being reviewed. So this helps ensure that there isn't any conflict of interest in the QA process. Additionally, individuals involved in QA should be appropriately qualified and experienced. Let's move on to reporting. It's important to ensure that there's clear responsibility for data reporting. And this includes, again, having a named person responsible for coordinating the analysis and reporting. It's particularly critical when the lab needs to report anything that's anomalous, that could potentially have an impact on the safety and well-being of a trial participant. Bear in mind, GCP, the #1 priority is protecting the people who are involved in the trials. The lines of communication between the lab and the sponsor of the trial or their representative need to be established and documented before the study starts. Archiving electronic data. The lab needs to ensure that the data remains usable over a long period of time. That's a matter of years. Now, when you think about how quickly technology develops, you realize that this is quite a challenge. Now, how many people nowadays have got the necessary kit to play a Betamax videotape or a Laserdisc or an 8-track cartridge? Now, one approach in the past has been to physically retain analyzers or computer systems that we use to generate the data so that they can still maintain access to it. But this involves regular testing of the equipment to ensure it all works. And as parts stop becoming available, it becomes a real problem and prohibitively expensive. Another approach can be to copy the data to different storage media. But the shelf life of the storage media is sometimes not as long as is actually required by the legislation. For example, recordable CDs. The data still needs the programs to be available as well to be able to read it. A lot of programs, as they're updated, aren't necessarily as backwards compatible as you'll need. And even when they are, keeping systems up to date can also be incredibly expensive. So these are all challenges you need to bear in mind. When the data is migrated from place to place in the course of archiving or system upgrades, there needs to be proactive planning and QC processes in place. Then I'll talk about archiving in general. So that includes digital and physical archiving. The responsibility for archiving of study and facility related data must be defined. And normally there's a dedicated archivist who isn't usually involved with the generation of data or the records that they're looking after. Documents from clinical trials need to be kept in line with the requirements of GCP and national legislation, like I said with the electronic data. These do differ slightly, but in practice it can mean indefinite retention. So the records retained need to include both study-specific data and non-trial specific data, otherwise known as facilities records. The data needs to be kept so that a clinical trial could be reconstructed many years after it took place to provide evidence that the protocol was followed and that regulations at the time were obeyed. So the facilities for archiving need to be secure, and they need to be designed to accommodate the type of material that's going to be stored. So this includes any electronic data storage, as I said before. Confidentiality of the subjects on the trial also needs to be maintained. The facilities don't need to be expensive, custom-made archives, but they do need to meet those criteria I've already said, and as a minimum they should offer things like pest protection, fire suppression, security from flooding, and obviously restricted access. One thing to particularly bear in mind with lab work, there are some types of paper records that need special consideration, like things that are produced on heat-sensitive paper by thermal printers. Now, I don't know if you've ever dealt with those, but it's a known fact that these will fade over only a few years. So you must have steps in place if you're going to be dealing with these type of records that they can be safeguarded in a formally controlled way, such as making certified copies at an early point while they're all still legible. Only authorised people like the archivist should really be able to access these records, and there should be a data trail indicating who's accessed what in the archive, and it should apply to paper and electronic archives. If ever materials are removed from the archive for whatever reason, there must be a booking process in place to ensure they go back in promptly and to check that what's returned is complete and intact. Now, the analysis. Now, I'm splitting the analysis into the planning stage, what you do if there's deviations, security, storage, analysis, and reporting. So first of all, the planning stage. It's preferable for a lab to base all of its work on the full clinical trial protocol and any amendments. If this isn't possible, as a minimum, the lab needs to have access to the sections of the protocol that are relevant to the work they're going to be carrying out. This is an absolute must. The lab must be kept up to date with the current version of the protocol, and if there are any amendments, they need to receive them as they're approved. And a mechanism for this information flow needs to be agreed with the sponsor or their representative before any work begins. The lab should draw up an analytical plan or work instruction before they start any work, and the plan should provide the level of detail that they need to actually perform the work. So to achieve this, it needs to contain things like the exact methodology they're going to be using, sample shipment and preparation criteria, reporting routes, timeframes, quality assurance activities, all that kind of level of detail. If there are any conflicts between the clinical trial protocol and the lab's own standard operating procedures, they need to be identified as early as possible and clarified before any work starts. Ideally, the sponsor should go through this work, the analytical plan, and certify that it meets the needs of the clinical trial protocol. So all activities on a study need to be underpinned by written policies and procedures. And years into the future, you might need to show that the analysis was conducted in the right way, in a way that assures its quality. So the actual activities need to be documented, not only through standard operating procedures and plans, but also through records that are captured at the time of the activity. The types and numbers and timings of samples that are going to be taken ought to be confirmed through the protocol and analytical plan, and any special handling that they might need because of the types of samples you're dealing with have to be built into the process. So this might include non-standard preservatives, protection from light, fast processing, things like that. So how do we deal with deviations? There must be procedures to quickly and effectively alert the sponsor of any serious deviations from the protocol or any other agreements. There needs to be enough information so that the sponsor can evaluate the significance of the deviation, especially in terms of patient well-being and integrity and reliability of trial data. Oops, I'll start that again. Deviations. There must be procedures to quickly and effectively alert the sponsor of any serious deviations from the protocol or other agreements. There should be enough information so that the sponsor can evaluate the significance of the deviation, especially in terms of patient well-being and integrity and reliability of trial data. If it's a deviation from the lab's own standard operating procedures, that also needs to be documented, including any remedial action. If the deviation could affect the integrity of data or subject confidentiality, things like that, the sponsor needs to be told immediately, possibly as well as the investigator. If there's no impact on trial data, the sponsor need not necessarily be informed, but it should still be documented. It should also be always followed through as part of the quality system to assure it doesn't recur. The records should always provide an audit trail, so decisions on whether or not to involve the sponsor can be traced and also justified. The reporting lines for deviations need to be clarified, and where the lab is part of the sponsor organisation especially, so the department acting as sponsor is kept up to date with the appropriate level of information. So what about serious breaches? The sponsor of a clinical trial should notify the licensing authority in writing if there's any serious breach, and this includes breaches of the conditions and principle of GCP in connection with a trial, or breaches of the trial protocol. And they need to inform the licensing authority within 7 days of becoming aware of that breach, which is why it's so important to have the information flow from the lab to the sponsor in a very efficient and controlled way. A serious breach is something that's likely to affect to a significant degree either the safety or physical or mental integrity of subjects on the trial, or the scientific value of the trial. The judgment on whether a breach is actually going to have a significant impact on the scientific value of a trial really depends on a whole range of factors, like the design of the trial, the type and extent of the data affected by the breach, overall contribution of the data to key analysis parameters, the impact of excluding the data from the analysis, things like that. But it's not the responsibility of the lab, it's the responsibility of the sponsor to assess the impact of a breach on the scientific value of a trial. So a few pieces of legislation that affect what you do with serious breaches. In UK, the guidance on notification of serious breaches was last revised in 2019. And it includes a recommendation that the sponsor notifies the chief or principal investigator of breaches which the MHRA have been notified of to enable implementation of CAPA, corrective and preventative action. The EMA's got a similar requirement. The sponsor has a responsibility in the UK to notify the MHRA using the approved form, and there's an image of it there. This notification can be delegated to the CRO, Discussions over who will do it mustn't delay reporting. So all of this needs to be agreed again before any work takes place. CRO lab should be assured that the sponsor has reported to the MHRA. So although this is very much UK and Europe focused, similar rules are going to be in place wherever you're working. Somebody's just put a question in, which is great. One thing I forgot to mention at the beginning is if you do have any questions, please put them in the chat box. And then what I'll do is I'll try and answer as many as possible at the end. Okay, thank you. Right, so to continue. Here's an example of a couple of things that would be considered to be serious breaches. Inadequate QC of interim safety reports used for dose escalation. And that's a serious breach because it has the potential for stopping criteria to be missed. Another one is a cohort's blood samples being invalid because they were processed incorrectly. So the knock-on effect is that one of the secondary endpoints couldn't be met. Therefore, there needs a substantial amendment required to recruit more subjects to meet that endpoint, and this means that inevitably some of the subjects are going to have been dosed unnecessarily. Let's look at security. As I said before, those who attended our webinar on good distribution practice heard about the measures that need to be put in place to protect the integrity of medicines throughout the journey, right from manufacturer to patient. So in GCLP, there's a similar process, but you're protecting the integrity in the journey from patient to lab. So I've split things into originator, transporter, and receiver. In terms of the originator, samples need to be labelled so they can be unambiguously identified, and there needs to be a system in place to track samples from their arrival to analysis or evaluation. And in terms of the transporter stage, just like in GDP, samples need to get from their source to the lab in such a way that their integrity and viability remains unaffected, whatever the distance and timelines involved. Again, like GDP, particular attention needs to be given to how long the samples are going to be in transit, conditions that ambient shipped samples might be exposed to during transit, temperature monitoring, etc. If the samples are frozen or refrigerated, there needs to be constant temperature monitoring, possibly with printouts given to the lab on delivery. And any requirement for this is another detail that should go into the analytical plan. So in terms of the receiver, on arrival at the laboratory, there needs to be some sort of booking in process. Samples should check that staff should check that the samples are in good condition. They need to be properly labelled, and they need to match the paperwork. Any deviations from this need to be investigated as soon as possible, usually by contacting the clinical site or the investigator as soon as the problem is spotted, or perhaps contacting the sponsor. Samples shouldn't be analysed until they've been positively identified and any deviations resolved. The only time you should carry on and analyse an unidentified sample is if a delay in analysis might cause a loss of sample integrity, maybe through stability issues. But if this is the case, you should do the analysis and then quarantine the result until the sample identity can be confirmed. So, in terms of storage, all clinical samples need to be stored under the required temperature and conditions. These need to be specified in writing. This is regardless of whether they're at the investigator's site before shipment or in the lab after receipt. Storage conditions can be specified in the clinical protocol, analytical plan, or other supporting documentation, but in all cases, the storage method for each sample and analysis type must be clearly described. The lab needs to have enough capacity, and I said this right at the beginning, enough capacity to store all the samples in the correct conditions, and that includes backup capacity, just in case something like a fridge or a freezer breaks down. And the provisions for this backup need to be clear and defined up front so that staff who have to deal with disaster recovery know exactly what to do. The storage facilities need to be monitored so they maintain the correct temperatures, and the monitoring records need to provide evidence that the samples are going to be still fit for purpose. Acceptable parameters for fridges and freezers need to be set and monitored. And what these parameters are is going to depend a lot on the individual situation and the scientific justification. But whatever it is, it should be documented and then implemented consistently. There should be clear procedures to cover actions if these limits are breached. Both the monitoring records and any actions taken in the event of excursions need to be documented and retained as part of historical records. Monitoring equipment like thermometers or automated alarm systems need to be checked and calibrated periodically, just like you do with all other equipment. So any analysis that's being taking part or evaluation work needs to use appropriately validated methods. The only exception is where one of the objectives of the clinical trial is actually evaluating the analysis method that you're using. But by and large, any analysis needs to use a validated method. The primary objective of validation is to ensure that the method is going to be robust, reliable, and reproducible. So in other words, not dependent on one technician who's able to make it work or needing to be routinely run 3 times before the results look acceptable. Routine system suitability tests, such as the use of QC samples, ought to be done as standard during the analysis. The nature of these tests and the acceptance criteria, again, define this as part of the validation and then implemented consistently before you start any work. I'm going to say this again, and I've said it several times, both the equipment and personnel should be fit for purpose, and you need to consider any factors that could affect the results. The criteria identifying when an analysis can be repeated should be defined and made very clear in the SOPs or materials. There should be no repeat analysis unless these criteria are met. It is vitally important that there isn't any selective reporting of results, and that where data are repeated or removed as outliers, you can justify the reasons, and that their reasons are transparent and documented. Quality control checks need to be an integral part of the lab's activities. What they are, their frequencies and so on, should be included in the laboratory's SOPs and methods. And examples include the use of QC samples, excuse me, in analytical runs, checking components in clinical kit preparation, manual data entry checks, secondary tube labeling, checks on calculations, things like that. There are a vast number of quality control steps that could be taken, but the overarching requirement is that quality control activities need to minimize the risk of mistakes. For this reason, it might be appropriate to take a risk-based approach when you're deciding on the type and frequency of checks that you're going to do. QC checks, like QA, most effective when they're performed by a second person, or some QC checks are obviously done automatically by the equipment itself. And this is because you don't want a technician checking their own work. The records need to capture the fact that a QC operation has been performed, when it was performed, and who did it. So the analytical plan, contract, things like that, all need to specify the timelines, method, format, and frequency of reporting. This should include any intent to provide interim reports. In general, there's 2 types of reports that the lab might produce: single time point reports that are per subject for a specific set of results. For example, microbiology on day X for subject Y. Or a combined analytical report that has multiple sets of data, interpretation of the results and conclusions. So something like a pharmacokinetics report. Whatever the style that's going to be used, the content needs to be complete and accurate. So again, it's a good idea to incorporate QC checks. Where data is sent on an interim basis, for example, before dose escalation points, the interim data sets need to be retained so that the basis on which decisions were made can be verified later if necessary. So let's talk about the patient, and let's talk about safety, confidentiality, consent, and blinding. As I said before, patient safety is the #1 priority, overriding all aspects of clinical trials, and this forms the cornerstone of good clinical practice. So it's vital to clearly define the lines of communication for reporting any issues related to subject safety to the sponsor and investigators. Issues might include the reporting of things like unexpected results or those that are outside the normal range, as well as any significant deviations. You might need to have a process for expedited reporting of reports. So this again needs to be considered before the study starts. Bear in mind things that could stand in the way, such as differences in time zones between the lab and the sponsor, upcoming holidays, etc., etc. A clear example would be an instance where safety blood alert limits are met or exceeded. There should also be a mechanism in place for things that are slightly less obvious, such as unexpected values in pharmacokinetic measurements. Anomalous results here might indicate misdosing or an important metabolic issue. There might be situations where lab data indicate a subclinical problem, even though there aren't any observed clinical signs. All data should be reviewed by an appropriately qualified person to identify things that are anomalous or out of range, and this should happen very promptly after analysis. And for the reasons I've already described, it's important to report things like this to the sponsor, even if the samples are actually analysed weeks after they were received. So the emphasis here is on swift reporting of issues once the results are known, regardless of how long it's been after the sample was taken, as it could still have a bearing on patient safety. ICH GCP, as well as safety, it grants every subject on a clinical trial the right to privacy and confidentiality. This means that the full identity of the trial subject shouldn't be revealed anywhere other than at the location where the trial is being conducted. So no subject identifiers should be received in an off-site lab. Now, obviously, we're all human, mistakes can be made, and occasionally samples may come through with information on them that could identify the subject. So what do you do then? Well, there needs to be a process in place in the lab that defines how you deal with samples and a requisition forms that contain information that compromises the subject's right to privacy. So this information, the information that identifies a patient, needs to be masked, but it needs to be done in a way that doesn't obliterate the other information that might be needed to identify the sample during the analytical process. If there's no other way of identifying the sample when you receive it than the personal details, these should be masked, and you should put in a unique lab identifier assigned to that sample in order to identify it. If something like this happens, report it to the sponsor or the investigator as soon as possible to stop it happening again. Excuse me. If a study is blinded, the integrity of the blind ought to be maintained. The protocol should specify who will be unblinded during the course of the study, and of course the legislation requires that that protocol is followed. If the lab's blinded and isn't provided with any randomization code, there's not normally any problem. However, labs are sometimes unblinded, for example, for pharmacokinetic analysis. In this example, it's common not to analyze samples from subjects on the placebo, and so analysts may be provided with randomization list in order to select the correct samples for analysis. There should then be a recognized process to deal with this whilst protecting the integrity of the entire study. Patient identity needs to be kept from others in the lab, so randomization codes must be stored and adequately controlled to ensure they can't be inadvertently seen by anyone else. This can be done as simply as putting them in a safe or putting in a locked drawer, or through the control of access if these are electronic codes. Think about reporting the results in a way that they can't inadvertently lead to unblinding. For example, pharmacokinetic results could be a dead giveaway, but other results that are significant and may lead to inadvertent unblinding, like a drug's known effect on liver enzymes or blood cell counts, need to be borne in mind. So in establishing the analytical plan, you need to think about these aspects and the appropriate steps that you will be taking to protect the blinded nature of a trial. So, and yet another aspect to think about in the analytical plan. Third aspect of GCP that relates to patients is informed consent. As with patient safety, these are ethical principles that have their origins right back in the Declaration of Helsinki. The informed consent documentation is approved by the ethics committees before the studies start, and the consent form is a controlled document, so that any subject involved in a trial needs to be fully informed by the investigator or their delegate, and the consent form needs to be signed by the subject before they start participation in the trial. By signing a consent form, the subject is consenting to the procedures outlined in the form and no others. So if you might need to do further tests, there should be additional consent. If you need longer term storage of samples than is on the original form, again, you may need additional consent. And finally, if you want to use the subject material for after the study for research purposes, again, you should seek additional consent. Carrying out additional tests in the laboratory without the patient's knowledge is a serious breach of their rights and GCP legislation. Sponsors must have agreed to the analytical plan, and that as they can then see what the laboratory intends to test for, and they can make sure it matches the protocol. But I've been talking about informed consent forms. Labs shouldn't see the informed consent forms because, of course, they identify the participant. There needs to be a process in place to inform the lab if consent is ever withdrawn by a subject at any stage, and to indicate how the lab should act if this happens. What they should do in terms of further analysis of existing samples, maybe what to do with data that's already been generated. Each time this arises, the subject should be asked by the sponsor how they wish to proceed on these particular points. The withdrawal of consent process is something that can be covered in a standard operating procedure, in the contract, or again in the analytical plan. Okay, so this is the end of the main part of the webinar. What I'm going to touch on briefly now is how GCLP has been impacted by COVID-19. So the MHRA, for example, has largely replaced its on-site face-to-face monitoring inspections with remote inspections. There hasn't been a lot of feedback so far, but there's been no horror stories, so the process seems to be broadly working. However, any significant issues with premises, activities, or management of test facilities need to be reported to the MHRA by email. So, how about maintenance and calibration of equipment? Well, if an engineer can attend a site to do either of these, that's fine, but you need to do a risk assessment before they do so. If the engineer is only available by phone or video call, a risk assessment could be okay to see if local staff can be talked through the process. If an engineer is not available to attend the site and remote supervision isn't possible, you need to do a change control or deviation and a risk assessment. It's possible that you could get equipment calibrated off-site, but if you're going to do that, do a risk assessment before any equipment is ever sent out. Another possibility is substituting equipment with other equipment. This could be okay, but bear in mind you'll need a risk assessment and you might need to revalidate the equipment. What about quality assurance? You need to assess the impact of reduced inspection. Possibly remote surveillance might be an acceptable alternative to face-to-face monitoring. In terms of remote document approval, validated system using electronic signatures should still be used if at all possible. Email approval of low risk approvals only is okay. So that could be for things like controlled document up versions, things like that. Paper docs can be printed, signed and scanned back remotely. But if you're doing so, make sure that the signature page is clearly identifiable for the specific document. But you can even do things like take photographs of documents on your phone as long as they're legible if no scanner is available. Okay, well, thank you for that. I'm running a little bit over time, so I have got time to answer one question. There are a couple of questions have come in, so if I don't answer your question, then what I'll do is I will email you separately with an answer. Somebody's asked, what about labs producing clinical kits? Okay, well, some labs are involved in producing kits that contain the sampling materials that are needed for the individual subjects on a study. This has got a number of benefits, and one of the main ones is, from the lab's perspective, you can guarantee the accuracy and quality of the materials that are being used. So it means that you've got more control over the quality of the samples you're going to receive. So where you're preparing these kits, you need to, as usual, have suitable processes in place to make sure the kits are prepared correctly for each study. Lab and the sponsor needs to agree exactly what the kit's going to contain, along with things like the shipment details and the number of kits you're going to produce. Yet again, it should all go in the analytical plan. So in terms of what the lab's going to need, they need large enough areas for the preparation of the kits so it can be separate from other activities. As I said before, it could be separated in time. So it could be that those kits are produced before the lab starts analysing samples. They also need to be able to monitor the environmental conditions to make sure that the kits are fit for use when they're prepared. And the individual components are being stored in the right conditions, such as preservatives and things, to keep their integrity. The other thing that the lab needs to do is it needs to keep an eye on expiry dates. So you need to make sure that the test kits they're sending out have enough shelf life to be usable. Now, for those who attended the GDP webinar, again, a lot of this is going to be quite familiar. So you need procedures in place that define the distribution, resupply, and you also need recall activities in place so that if anything is found to be wrong with these test kits, you can get them back quickly and effectively and resupply them at relatively short notice. The kits need to be held at the sites and they need to basically remain in date for however long you think they're likely to be held. Okay, right, well, that's the end of the webinar. Thank you so much to everyone for attending. Just a quick recap. I said at Whitehall Training, we've got a lot of online training courses. They're supplemented by face-to-face and blended learning solutions, if that's what you want. If you decide you want to go further with GCLP, we do have a full online course on the subject. And as a thank you for attending this webinar, I will be emailing all of you with a copy of the recording, but I'll also email you a special code that will give you a 50% discount on any of our courses that you can enter at the checkout, and that discount code will be effective up until the end of this month. If anyone's interested in ordering and they want to be invoiced, or they want to discuss things like corporate discounts, you can email me on the email address on the screen, ashley.smith@ infonetica.net. Okay, well, thank you very much for attending again. We will be running some more webinars, probably on a 6-monthly cycle. So I will contact you again when we're going to be running webinars again. But really, it just falls to me to say thank you again and stay safe. Goodbye.